All right, so today I'm going to start chapter 17, which is entitled From Gene to Protein. In other words, we're going to be discussing how we take DNA and turn it into a protein ultimately, which is what you would like to get from uh, DNA. That's what its job is to make proteins ultimately. That's what it holds the instructions for. So what I'm going to do over the course of the next couple days is I'm going to split this chapter up into like three different smaller lectures. Today's going to be a little bit more cumbersome. There's going to be a lot more we cover today, which is going to talk more generally about the process of going from DNA to a protein. And then we're also going to talk about transcription and RNA processing today. And the next one, we're going to cover translation, which is the creation of the actual protein. And then finally, the last one is going to be discussion of mutations, because all this stuff kind of is linked together, but it would be an hour something of listening to this over and over again. So I'm going to break it up into like three different ones. All right. So right off the bat, we are going to talk more generally about the process behind going from DNA to ultimately to a protein. That involves two major processes known as transcription and translation, which we see right up here. Transcription is the production of RNA, specifically mRNA, under the direction of DNA, whereas translation takes that mRNA strand that's produced in transcription and turns it into a protein. Okay, and so we see this down here. Right now, we're going to disregard the prokaryotic cell and just kind of look at the eukaryotic cell that we see down here, because that's the kind we have. We are eukaryotic. So what we see here, we see DNA. We start with our ultimate architectural plans for our entire body in the DNA. That DNA undergoes the process of transcription, which we see here, and ultimately turns into what they call pre-mRNA. We'll talk about why they call it pre-mRNA a little bit later. But this pre-mRNA goes through RNA processing, where it finally gets its certificate, if you want to say that. It is now actual mRNA. Okay, so now at this point here, what we actually see is that the nuclear envelope separates the two processes of transcription and translation. Transcription happens inside the nucleus, whereas translation happens outside. So the mRNA actually has to leave the nucleus for it to be translated. But during the process of translation, we see that the ribosome is one of the primary organelles that is going to be utilized to take that mRNA, make heads and tails of it, and create what is known as the polypeptide or the protein strand. Okay. One of the ways, I know most of you guys know this already. This is all kind of review. But if you do have trouble with it, it's a fairly simple analogy that a lot of you guys can grasp onto at this point. Um, a lot of you guys are in the process of going through and applying for colleges, for acceptance into colleges. And most of you guys, if you haven't already, will figure out that you have to send your transcripts um, to the college, right? So that's one way I always remember this, is that the DNA is your intelligence. Okay, the DNA is your intelligence, and we've got to get that intelligence into a really, really good school. Let's say it's Harvard, right? And to get that intelligence into Harvard, we have to somehow portray to the Harvard board of admissions board that you are smart enough, and ultimately we want to translate your knowledge into college acceptance, right? So what we've got to do is take your knowledge and we turn it into transcripts. And your transcripts are basically just a printout of how smart you are, right? On your transcripts, you've got all of the classes you've taken, you've got all of the grades for the classes you've taken, you've got all the credits there. And so the transcript is a kind of a throwaway copy of how smart you are. Like, you wouldn't want to throw away your entire brain, you'd die, right? So instead, we create this transcript, in other words, mRNA, that can be sent to the colleges to say, this is how smart so and so is. All right. At that point, the colleges get your transcript, your throwaway copy of your intelligence, and hopefully we want them to translate that into an acceptance letter that they send back to you. So that's what we see here. They look at your transcript, which is exactly what's happening here. You can think of the ribosome kind of as the admissions board, and they look at your transcripts, and in the end, they hopefully spit out an acceptance letter, which is like your polypeptide. Okay, so hopefully that helps you. It always helps me because I always associate that term transcription with transcripts. Okay, so use that if you like it, great. If not, no worries. Figure out your own way. Anyway, if we're going to discuss how we take DNA and turn it into a protein, we're going to do it step by step. So we have to talk about the genetic code first and what it means. The question at the top poses, how are the instructions for assembling amino acids into proteins encoded into our DNA? One thing we know for sure is that our proteins, or polypeptide strands, are made up of amino acids. And we know for sure that there are 20 different amino acids. Now we got to take that into account. We know that DNA ultimately turns into proteins, but we have to figure out, scientists had trouble figuring out, okay, what kind of association did the DNA have with creating the protein? So we know there's 4 nucleotide bases in DNA. There are 20 amino acids. What is the relationship there? What's the correlation? And it turns out that there has to be multiple bases that will ultimately code for a single amino acid, because we can't have 4 DNA bases, one for each amino acid, because there's 20 amino acids, just not enough. So what scientists ultimately figured out is that a sequence of 3 nucleotide bases on the DNA corresponds to each amino acid with some redundancy built in. And we'll talk a little bit about that redundancy being built in a few slides, because that can become important when we discuss why there's multiple 3 base pair sequences that code for the same amino acid. So we will talk about that redundancy, but ultimately what scientists settled on was the fact that 3 nucleotide bases, A's, T's, C's, or G's, together in a grouping of 3, corresponds to the creation or the recruitment, ultimately we'll call it the recruitment, of an amino acid to be put onto the growing polypeptide. Okay. Now, the DNA strand, we know that there are 2 DNA strands, obviously. We learned that last chapter. But only one of those 2 DNA strands is actually transcribed. Okay. Now, the strand that is transcribed is called the template strand. Okay, the template strand, and we can see that one down here. For some reason, this template strand right here is the one that is looked at by to create the RNA and then ultimately to create the protein. The one that is not used, they call the coding strand. And so for some odd reason, they call the one that's not used the coding strand, but they do call the one that is used, they call it the template strand. So do keep that in mind, that when we are talking about the template strand of DNA, it is the one that is looked at by what's known as RNA polymerase, and RNA polymerase will make the RNA. So it is the template strand that ultimately creates the mRNA strand. That's the code that is looked at to make the mRNA. Okay? Little formatting issue here when I took it from PowerPoint to Google Drive. But anyway, it says which DNA strand is chosen as the template depends on the gene being transcribed. So it's not always like just one side of the DNA strand. It could be the other side, and it all depends on which gene it is. That's not something we're going to get into here today. A lot more complicated than I really have time for right now. But that is one thing I want you to understand here, is that even though we do have 2 DNA strands, only one of them is actually utilized in the transcription process. Okay. Now let's talk a little bit more about those base pairs that we mentioned before. Something known as the triplet code. The triplet code is the genetic instructions for a protein chain that is written for in the DNA. Okay, it's a series of non-overlapping 3 nucleotide words. And something known as the codon is the mRNA version of the triplet code. Okay, so we talked about 3 DNA base pairs ultimately is going to be looked at when we're creating the protein in the end. And those 3 DNA base pairs are transcribed into what is known as the codon. Okay, the codon is the mRNA version of that triplet code. So I'm going to use that term codon quite a bit here when we discuss what ultimately mRNA is, how it's created, and then how it's translated in the end. Okay, so the codon is created by matching with the DNA triplet code. Okay? The mRNA strand that we ultimately see will be complementary to the template strand. And that's what we're looking at is the DNA triplet code, let's say, of AAG. Okay, so let's say the triplet code AAG. So those are the three base pairs of our DNA, for instance, AAG. The codon is going to be complementary to AAG, all right, and it would be UUC. Okay, and why do I say UUC? Because there are no T's in the RNA, right? So if we were just going to look at the complementary DNA bases of AAG, it would be TTC. But since we're talking about creating an mRNA strand from that, it's actually going to be UUC. All right, a little bit confusing. It helps with pictures, but we're going to take a look at it. All right, so we see right here, we've got our DNA template strand. This is the strand that we see up here of gene number 1 that is going to be read, and it is going to ultimately, once it's read, it's going to create the mRNA strand. Now, here we see this first triplet code, okay, ACC, the first triplet code here. Once we read it and create mRNA, the complementary codon is going to be UGG. If we were creating complementary DNA strand, this would be TGG, but since it's RNA, we know that this U is going to take place of the T because RNA does not have T's, does not have thymine, okay? We look at the second triplet code, AAA. If we're creating the mRNA codon, it's UUU. Again, for the same reason, there are no T's in RNA, so we replace the T's with the U's. Okay? And ultimately, this is what happens during transcription. We take that DNA template strand, it is read, and we get a complementary mRNA strand that is based off of it. Okay? This mRNA strand is translated to get this amino acid strand, which we see right here. Okay? On we move. Now, when we look at the mRNA strand, or when our cells look at the mRNA strand has to be read very specifically, okay? Each mRNA codon corresponds to a specific amino acid that is ultimately put into the chain in a correct order. These amino acids have to be connected together in a very specific order if you want a properly functioning protein. That is something we've talked about a lot, especially the thing all the way back to chapter 5. You have one messed up amino acid, you have one messed up protein, okay? You change one of those things, you flip-flop the order of one of them, you have a defunct protein that doesn't work. And we're going to talk about some extreme examples of that when we get to the mutations. So to attain this specific amino acid order, the codons must be read in the correct, what is known as the reading frame, or the correct groupings. There's a lot of things that can change that reading frame, including mutations of many different varieties, which we're going to ultimately talk about at the very end of this lecture series, this 3-lecture series. Okay. Now, the reason I went through all of that is because I wanted to introduce this table that you see right here in front of you. You guys have probably seen something very similar in like a pie chart or a circle chart. I really like this one. This is the one I always use. But the pie chart works just the same. But what we see right here is a layout of all of the different codons. Okay, so the different codons here, these are like UUU, UUC, and also next to them, the amino acids that they code for. Okay, so this is what we would consider to be called the genetic code. There's 64 codons that code for 20 amino acids. Now you'll notice, as I mentioned before, that this code is redundant, okay, but not ambiguous. So meaning it's redundant for a reason is basically what it means. So redundant meaning you've got 4 different codons here that code for the same amino acid. That's redundancy, okay? In some cases, it's because it's a fail-safe, all right? And for other instances, it's because when we look at these last letters, those are the only things that really tend to change here, okay? And there's something to be said for that, because if we have some kind of mistake happen, maybe in this last letter, there is some wiggle room. And in fact, they call it the wobble effect. There's some wiggle room so that you can still get the amino acid that is supposed to be there. So in this case, proline, all right? So we look at the reading frame. Okay, this is if we were to look at this uh, right here, okay, it doesn't make a whole lot of sense. It's got improper reading frame. If we read it t, her, ed, oga, tet, heck, at, it doesn't make a whole lot of sense, right? That's because we've got the wrong reading frame. Now let's shift that reading frame and we read it this way. The red dog ate the cat. Okay, now it makes a sentence that actually makes sense. This sentence up here makes no sense whatsoever. Now we change that reading frame down here, and we do different groupings of 3, and Clifford the big red dog ate the cat. Okay, so that's how important reading frame actually is for all of the reading of the DNA to get the proper protein. All right, so skip that. We're going to get a closer look here into the process of transcription, and this is where we're going to kind of speed through this stuff a little bit. I'm not going to spend a whole lot of time on this because a lot of it is just spelled out for you in the slides themselves. But transcription is the DNA-directed synthesis of RNA. Now, the primary enzyme in this entire process is known as RNA polymerase II. If you remember back to chapter 16, we had what was known as DNA polymerase I and DNA polymerase III. You know, try, maybe some of you are like, where's DNA polymerase II? Where is it? It's gone, it's missing. Well, it's because they named RNA polymerase II. So RNA, they didn't want to mix everybody up. So there is no DNA polymerase II. It's because we have an RNA polymerase II. But what RNA polymerase II does is it pries the DNA strands apart. Remember, we're reading the DNA strands, so it's prying the DNA strands apart, and it hooks together new RNA nucleotides. So in effect, if we're trying to relate this back to what we saw in chapter 16, it is very similar to DNA helicase, okay, the one that unzips the DNA, and DNA polymerase I, all wrapped up into one enzyme. That's what this is. It's like helicase and DNA polymerase I. Okay, so it can do multiple things together. So the DNA sequence where RNA polymerase attaches to is called the promoter region. This is the starting line for transcription. Okay, and the stretch of DNA that is being transcribed is known as the transcription unit. In other words, we could think of this thing as the gene. The transcription unit is the thing that is being transcribed to ultimately create a protein in the end, and that's what genes are. So that's what we're gonna basically call the transcription unit a gene. Okay, so we see here this translucent guitar pick looking thing right here is RNA polymerase. This area that's in kind of minty light green is called the promoter region. And then we have this little start point. This dark blue area right here is called the transcription unit. For our purposes, that is also known as the gene. Okay. Now, what we see happen at the very beginning in this process is known as initiation. Okay, that's where we actually have the pardon me. The RNA polymerase will associate. The RNA polymerase will actually associate with the DNA strand. Okay, so we see that happening here, and we start to see the RNA polymerase do its job. It's starting to unwind and unzip the original DNA strand, and it's actually already started the RNA transcript down here we see in red. Okay, let's get a little bit more detail about initiation phase. Okay. In the initiation phase, it's more than just the attachment. There are more things that happen in this phase than just the attachment of the RNA polymerase. In fact, what we see here is the RNA polymerase will actually attach to the promoter region at a specific place called the Tata box, which is one of the most fun things to say in biology. The Tata box signals the initiation of RNA synthesis. The top box is kind of like the starting line. Okay, it is a portion of the promoter region that the polymerase will attach to specifically. Okay. In addition to this promoter region and the top box within it, there are what are known as transcription factors. And transcription factors are like accessory proteins. They're like little proteins that will bind to the DNA, and it will be like a docking point or a docking bay for the RNA polymerase to attach to. And they're very easily seen here in this picture. All right, so let's start at the very beginning here. Here is our stretch of DNA that's ultimately going to be transcribed and turned into a protein. Here in this kind of light lime green area, this light lime green area here, we've got the promoter region. Right here we have the tata box, and all that is a stretch of TAs in the DNA genetic code. Okay, it's TA, tata box. And the tata box is the area where the transcription factors, that's what these little purple bubbles are. That's where they will start to attach to. Okay, so the transcription factors, these little proteins, will attach to the tata box right here. And what we notice is that the RNA polymerase uses those transcription factors as a docking point, as a beacon to say, this is where you need to set up, this is where everything's going to start. Oops. And once the RNA polymerase attaches to this point right here, then it starts off at the starting point. All right. Phase 2 of this process is known as the elongation phase. And its elongation phase is very, very simple. We just have to know that during the elongation phase, RNA polymerase moves along the DNA, untwists it and unwinds it and unzips it as it goes, and lays down new RNA bases. Okay? So, transcription progresses at a relatively fast clip, about 60 nucleotides per second. You don't need to understand that or remember that for a test. But one thing we will notice, and I'm going to get to a close-up picture of it, is that the RNA, when we create this new RNA strand, is put together in almost the exact same fashion as the DNA replication we talked about in chapter 16. Okay, so we look at this RNA that's been created here. We notice that these new RNA nucleotides are being attached to the growing 3' end. In other words, the RNA transcript that's being created here is growing in a 5' to 3' direction, which is exactly how we would create a brand new DNA strand. Now, in this case, there is no leading or lagging strand. It's just the leading strand. I shouldn't say there is no lagging strand. It's only the leading strand. RNA can be produced in a continuous fashion until it is created. Okay, so we see that the direction of RNA transcription we see is in this direction along with this arrow. Okay, so the DNA over here is going to be continued to be opened up by the RNA polymerase. As it's opened up, these new RNA bases are brought down. Okay, and we see them attached to this growing 3' end. Right, so this is the old, newly made RNA. This is the newer part of the RNA. All right. At the end of this entire process, we have the phase 3, which is called termination, and all that does is at the end of the gene or the transcription unit, there's going to be a sequence of bases that tells the RNA the RNA polymerase to go ahead and just drop off and be done. Okay. That terminator region, what they call it, can be different based on whether or not you're looking at a prokaryote or a eukaryote. But in eukaryotes, the polymerase continues transcription after pre-mRNA is cleaved from the growing RNA strand. Okay, so the polymerase eventually falls off of the DNA when the entire process is done. So we call this process, or this last phase, termination. Okay, really the big part you have to understand here is the initiation phase with the inclusion of the promoter region, the tata box, and the attachment of the transcription factors. And once you have the transcription factors, they are ultimately going to bind on the RNA polymerase. RNA polymerase then creates RNA by unzipping the DNA and making the RNA strand in the 5' to 3' direction. Okay. And section 3 gets into a process known as RNA modification. Okay, eukaryotic cells modify RNA after transcription occurs. That's why in some of the diagrams we've seen, they refer to the freshly created RNA molecule as pre-mRNA because it hasn't been modified yet. So there are enzymes in the eukaryotic nucleus that will modify the pre-mRNA before the genetic messages are dispatched or sent to the cytoplasm. Okay, so let's talk about what happens to the RNA before it is actually sent out to the cytoplasm for translation. One of the first things we have to talk about here is the modification of the mRNA ends, the ends of the RNA molecule. Okay, the ends of the RNA molecule are modified in a very particular way. The 5' end of the RNA molecule receives what is known as a guanine cap. Okay, and the 3' end gets a poly A tail. And in other words, what you're going to see at the 5' end is a little guanine. That's what we see here in yellow. Okay, the guanine here is what they call the 5' capped. It's attached right to the end, the 5' end of the pre-mRNA strand, which is seen here. Okay, at the other end, the 3' end, they call the poly A tail. Basically, all that is a giant long tail of adenines, okay, or A bases. So these modifications share several functions. First off, they seem to facilitate the export of the mRNA molecule. We mentioned already that the process of transcription happens in the nucleus. The process of translation happens in the cytoplasm. And these act as kind of exit tickets, okay? The RNA, after RNA processing has occurred, you get the guanine cap. It's like, hey, here's my exit ticket. I need to get out to the cytoplasm to make sure that I can be translated. Okay. Another thing that these help for is protection. What the cell does a really good job of when it sees something in the cytoplasm that it doesn't really know what it is, it does a really good job of chewing it up and making sure that it kills anything that is maybe looks foreign. And so what we see these things do is they act as like a buffer region. Okay, the mechanisms that chew up these foreign molecules oftentimes start at the end of the molecule. And if it's got this giant poly A tail, the hydrolytic enzymes are going to start there first and hopefully chew that portion up before it chews up any of the important stuff that we see here in red. Okay. It also, these caps also help ribosomes attach to the 5' end. Okay, so this little guanine cap is almost like an entrance ticket as well. The guanine ticket is an entrance ticket to the ribosome to make sure that it gets to where it's supposed to go, make sure that everything is oriented just properly so that translation can ultimately occur. All right, there's another process known as RNA splicing. RNA splicing is really cool because RNA splicing allows for a lot of flexibility in gene expression, okay? Most eukaryotic genes and their RNA transcripts have long non-coding stretches called introns. And these introns are things that are going to be basically cut out, okay? Now, the way I remember this here, there are introns and there are exons. The regions of a gene that get expressed, okay, and when you get expressed, you are turned into a protein, so you stick around. If you are expressed, you are an exon, okay? Regions of the gene that are expressed and turned into proteins are called exons, okay? That's what we see listed right up here. The process known as RNA splicing removes what are called introns, okay? And introns are, in some circles, are called junk DNA, okay? They do have a purpose, which I'll talk briefly on, but the introns in this case happen to be junk or unneeded, okay? So they need to be cut out. And so this next slide shows something known as the spliceosome, kind of another one of those really cool words. The spliceosome here, this big thing right here, is kind of like DNA scissors, and they specialize in cutting out chunks of unused or non-coding or unwanted DNA. Okay, and we're not going to go through the process of how this occurs, but spliceosomes are responsible for taking out those introns. Okay, so what is the importance of the intron? Well, in a process known as alternative splicing, some genes that have both exons and introns can change um, which are expressed. So in some circumstances, in some genes, the introns can all of a sudden become exons under certain situations, okay? And the exons can be introns. So everything can kind of flip. So what that allows for is a very, very large increase um, in genes, not genes, but proteins that can be created. Okay, the number of different proteins an organism creates is much greater because you have different ways of expressing the same gene. In some instances, stretches of DNA called the introns can be considered exons. So that's why splicing is so important. It allows for very, very large diversity and variability and flexibility in the expression of the genes. Okay. So before we jump into translation, I want to get into the different types of RNA. There's 3 different types of RNA that I want to make sure you understand, and they're listed right here. We've already talked about one of them, known as messenger RNA. This is your disposable copy of the very valuable DNA code. You want to be able to have a disposable copy so that you can keep a nice pristine copy in the nucleus in the DNA, right? So that's what messenger RNA is. It carries information specifying the amino acid sequences of proteins from DNA to the ribosome. Okay. The 2 that we're going to talk about more specifically in the next section are tRNA and rRNA. Both of those serve in the process of translation. tRNA translates mRNA codons into amino acids. It's kind of like a little UPS truck, and I'll explain that more when we get into the next section. And then rRNA is nothing more than the structural part of the ribosome. Ribosomes are actually made up of RNA. And so in some of the pictures that we're going to see, I will show you that. I will make sure that you understand that ribosomes are made up of RNA, and that type of RNA that they're made up of is called rRNA or ribosomal RNA. Okay, so I wanted to introduce these before we get into the next section, which I'm going to make a separate video on.